So welcome to this series of video conference organized by the Partners Club of the GDR Imabio. So it's a pleasure today to organize this session with Telight. And this session will be moderated by Serge Monnet, who is a CNRS researcher at the Fresnel Institute, and also Deputy Director of the GDR Imabio. So I stop my sharing. Zuzana, if you want, you can share your desktop. Okay, so just to inform everybody, you can we will have the two seminars from Zuzana Nováková from Telight and Vaclav Bočan. And so then you can ask questions in the chat part if you want, and also in live at the end of the two seminars. And I just remind you that the session is recorded. So if you don't want to have your face on the video at the end, so please do not activate your camera, but you are still free to ask questions in live at the end. So Zuzana, it's yours.
Thank you, Virginia. Good afternoon, everybody. Welcome to this Zoom webinar focused on the quantitative phase imaging technology. And thank you, Virginia, for giving us the opportunity to speak about this technology, to introduce it to a broader audience. My name is Zuzana. I work as an application specialist in the company called Telight. Telight is actually a startup based on the field of optics, and we focused on optical solution for lifestyle imaging, and we are actually located in the Czech Republic. So greetings from Brno. And today I'm here also with Vaclav, who is our key user at the Max Planck Institute in Dresden, who uses our microscope or our technology in his research that he will introduce later. So just briefly, let me introduce the technology of quantitative phase imaging, because even though it's been around for a few years, I think that not enough people have heard about this technology, and maybe, hopefully, you will also see useful in your research. So quantitative phase imaging, it's label-free technology, similar to the, for example, phase contrast technique, doesn't require any label, and it's non-invasive and non-disturbing, so perfectly fit for monitoring live cells. Compared to the phase contrast imaging, it's quantitative, which means that you are recording your cells quantitatively. You can quantify the dryness of the observed objects. It doesn't require any high illumination power, so it's of low phototoxicity, suitable for long-term lifestyle imaging. There is, of course, no photo bleaching since it's label-free technology, so it provides constant data over time, and it's really fast in the imaging. There is no difficult reconstructions, no scanning, just one single image from the camera plane, and you already see the image of your cells. And it's really of high contrast without any artifacts like scattering or some coherence noises can be sometimes present in this technology. And good quality of the data is one key condition for cell segmentation that we will also touch in this webinar. So quantitative phase imaging works in the principle of light interference. So in such microscope, the light is split from the light source into two beams. One beam passes in the object arm through condenser through the sample to the objective, and one beam passes only through the reference sample in the other arm. What happens when the light beam passes through a sample? You can imagine that the light is slightly retarded, slightly slowed down by the sample because of its optical properties. Compared to the reference sample, where just light goes through empty slide and it's not slowed down. And these two beams, they are brought together again at the camera plane, and they interfere. And what the camera sees as a digital recording microscope, the camera sees the interference pattern, which we call them holograms. And from this interference pattern of these two beams, one beam that passed through the sample and the second beam that passed through just an empty optical pathway, from holograms, we can actually reconstruct different types of images. One type of the reconstructed image can be, for example, amplitude, and because cells are transparent, so you know that in the amplitude image in classical bright field microscopy, you see really small contrast of them, but in the phase image, you see nicely the structures because cells are affecting the phase of the transmitted light, not its amplitude. So camera is seeing holograms, and then we numerically in software reconstruct phase image. That's how the technology got its name, quantitative phase imaging. So from these black and white stripy interference images, we can actually reconstruct the image of cells, maybe similar to the phase contrast microscopy. But the difference here, the key difference here, is that this technology is quantitative. So actually in each pixel, we can detect how much in terms of radiance in the units of phase, how much was the light delayed when it passed through the studied object. And so this image has a quantitative scale that can be expressed in the units of radians. But there is one more step that you can also use in this technology. You can convert radians into picograms of the dry mass. So you can quantitatively measure how many picograms each cell actually has. And this is the key application that Vaclav will describe how he's measuring and using this value in his research. What is the cell dry mass? Maybe a term that is new for you. Cell dry mass is all non-water content of the cell. In majority of the cells, it's mostly 70% is water and 30% it's this non-water content of the cell. Those are the biomolecules with the refractive index that is different from the water, because usually you image cells in the water-based media. So water is around, water is inside the cytoplasm, and the dry mass, the other biomolecules, are actually creating the phase shift. The dry mass consists mostly from proteins and lipids, but it's worth it to say that there are also other biomolecules that contribute to the phase shift, to the retardation of the light. It's mostly glycans, nucleic acids, for example, and other metabolites. Why is it even good to measure dry mass about living cells? Actually, dry mass can serve as a sensitive indicator of cell status, of cell cycle, the condition how the cells are growing, prospering, how it affects their metabolism. And of course, the cell movement is directly related to the measurement of the dry mass, because transferring the mass from one end to the other end is basically how the cell moves. Here are just some graphs to show you how the mass changes over time during different cell cycles. So you can see on the images that the cell, there are one cell, and then it shrinks and then divides into two cells. This is nicely seen also in the time graphs, where the cell mass increases, then it drops to the half, and then the daughter cell starts growing again. And the lower graph actually shows the reciprocal value, the density, which is expressing picograms per unit of area per micrometer square. And these high peaks in the cell density correspond to the moments in the cell cycle where the cell is most rounded, most dense, and is preparing for mitosis. And measuring the cell dry mass can also serve as a sensitive indicator in the toxicological studies, because it's a really sensitive parameter that can be affected by many testing conditions, by treatments, for example, some drugs. And if you are able to measure really with high sensitivity to measure the phase shift, which in our system you can measure really in milliradians, the changes of the phase, you can also convert these units to femtogram changes of the dry mass. So our system can measure really sensitively these changes of the dry mass over time. Microscope is called Q-phase. In basic principle, it's an inverted transmission wide field microscope. The difference from conventional microscopes is that it consists of two optical arms. So there is a set of condensers and set of objectives where the light propagates. In the object arm, we equip the microscope also with the fully motorized stage for performing multi-position experiments and stage job incubator to ensure the proper conditions for cells during the even long-term imaging. Yeah, this is the principle that there are really two optical arms in the microscopes. When in the object arm, the light is slowed down by the object, and in the reference arm, it doesn't. You may notice in the image that the microscope is also equipped with the whole set of objectives, so the system allows different application, from studying many cells in small magnification to going really into details and using high numerical aperture or oil immersion objectives. So different applications can be solved with such system. I know that I highlighted that label-free microscopy is good because it avoids affecting the cells and photobleaching over long-term experiments. But still, we know that fluorescent microscopy is really key in cell biology nowadays. So fluorescent microscopy is bringing the specificity into this imaging. So our microscope also allows to do correlative imaging between quantitative phase imaging and fluorescent imaging that allows you to see label structures or to confirm specific processes with some fluorescent probes. And microscope wouldn't be complete without software, actually. So we have the software that is designed for controlling the microscope to allow proper tuning of the microscope, setting up multi-position time lapses, to correlative imaging. And then in the software, we also allow users to perform segmentation of the acquired data, so and analyze them using different types of graphs and analytical tools. So actually, the microscope and the software is like complete package where you can start with the experiment and finish with the graphs and data already prepared for your publication. I think that Vaclav will also have some screen captures from the software, and he will maybe mention the segmentation also there. And what are the possible applications of such technology, which compared to conventional microscopy, really the unique feature here is that it's label-free, so it allows to monitor also sensitive cells like stem cells, developing cells, neural cells. So most of our application lies in the cancer cell research. We're monitoring cell proliferation or how to stop cancer cells from growing, how to stop them from how to prevent their cell division, but also their migration. So really large field of our application is in cancer research, but also this technology is suitable for drug testing because compared to the fluorescent microscopy, you study only the effects of the tested compounds. When you are avoiding any cell staining, washing, manipulation with the cells during the experiments, you can truly say that the effects that you are seeing are really coming from the tested drug. Neurobiology, mechanobiology. I could speak about the application of this technology much longer. So today we picked only one with Vaclav. At least I prepared a slide full of moving things, full of videos that were acquired using this technology. So in the upper left corner, I just put there some example of cell which is binucleated, full of vacuoles. Those are the bright structures inside. In the QPI and fluorescent imaging. Then going to the right, there is a video of the cells dying using necrosis. So you can also study cell death as a really distinguished process that can be nicely studied through cell dry mass measurement, cell proliferating even into really high confluent cells. This microscope can be also used for studying co-cultivation, so you can combine different types of the cells in the same experiment. You can study cells even in highly scattering media like collagen fibers, so these like 3D scaffolds where the cell is interacting with the collagens. In the bottom row, in the middle, those are neurons and growing axons and interacting with each other over many hours, and then some more videos of cell proliferating and moving, where you can nicely see also the intracellular structures. Because bear in mind still that these images are quantitative, so higher the intensity or darker the pixels, it means those are the parts of the cells where there's the most of the material. So usually nucleus, nucleoli, or some vesicles in the cytoplasm. And also you may notice in these images clear background. So there are no background artifacts, no scattering, so you can nicely distinguish the boundaries of the cells. And if you can nicely do this background discrimination, then the segmentation is much more precise, and you can achieve good segmentation quality using even simple watershed algorithms if you have such nice input data in the beginning. So they are just some examples. I hope that I got some fans for QPI label-free technology, quantitative phase imaging label-free technology. And if you have any questions regarding this technology in general, I'll fill them in the chat and I will answer after Vaclav's talk. So that's all from me, representing Telight, and now I will pass my word to Vaclav.
Thanks a lot, Zuzana.
Thank you, too. I hope you can see my presentation now.
Yes.
Good early afternoon to everyone. And first of all, thank you for inviting me to this webinar. I will be very happy to introduce you to my research. I'm Vaclav. I'm a PhD student at the Max Planck Institute of Molecular Cell Biology and Genetics in Dresden, in the lab of Rodenfels. And the lab is focusing on metabolic research from the thermodynamic point of view. And today I would like to briefly introduce you to the project I'm working on, which is dealing with quantitative phase imaging, of course, and is applying that in the thermodynamics of metabolism. The story is still being developed, so you won't get a full story arc from the beginning to the end, but at least you will get a flavor of how one can use QPI in scientific research. And the theme of today's talk will be getting specific, which I will introduce to you just later in the talk. So the reason why we are interested in metabolism in the lab is that metabolism is actually the key player in cell physiology. It is composed of many interlinked biochemical reactions, and it feeds into all the processes going on in the cell. For example, in proliferation, metabolism is supplying the building blocks to make a daughter cell. In signaling, many metabolites or intermediates in the cell feed forward into the biochemical interactions of proteins and signaling cascades like phosphorylation or other post-translational modifications. And last but not least, metabolism is also very important in cancer biology, as Zuzana has already mentioned, actually, because it's known for a very long time that in cancer switch, cells acquire new metabolic features or lose some other ones. So then, for example, the very well-known Warburg effect can take place, where cells abandon oxidative phosphorylation and go into glycolysis and fermentation, even though oxygen is abundant. And of course, we want to understand metabolism as well as we can, ideally through a comprehensive theoretical framework, which could be applied to very different modalities through which we can perceive and study metabolism. In ideal case, that should be quantitative to get really well comparable data from different measurements. And it should be a general framework, meaning that through the same framework, we can study very different model systems, not only cell lines and whole organisms, but also systems in different developmental phases like embryo development or aging, or even non-model systems far from phylogeny tree like microorganisms or non-model vertebrate. And last but not least, we should be chemical species and chemical pathway independent, because if we want to go general, we will be necessarily dealing with biological models with very different metabolic pathways, so that we should not be dependent on specific assays for specific chemicals, but rather we should strive for generality and broad traits which can apply to everywhere. And in the lab, we believe that thermodynamics is the answer to this problem. So thermodynamics, as a branch of physics, is driven by rigorous laws compared to biology. So we can rely on very firm theoretical basis, for example, the law of energy and matter conservation, which states that no energy or mass can be created nor destroyed. And the second law of thermodynamics, which states that the entropy in an isolated system is always increasing. And since thermodynamics applies to everything in our universe, it as well applies to metabolism. And from this, the key question of my project directly stems, which is asking how is it possible that growing cells can manage their all metabolic needs and grow, and they can obey these laws of thermodynamics at the same time. So before diving into that question, I want to briefly introduce how quantitative phase imaging can help here. Zuzana already told you everything important, so I can be really brief here. So in the lab, we are using the Q-phase microscope, the quantitative phase imaging microscope, which is just one kind of the QPI microscope because it's a very broad family of instruments with very different flavors. And in this microscope, I cultivate cells, living cells over periods of time. And I can acquire their growth in the microscope with supplied atmosphere and heating, and follow their growth for even up to several days. As Zuzana already mentioned, the perks of QPI and Q-phase specifically is that one can get quantitative phase imaging, which can be done on live cells, and that can be also combined with fluorescent modality. So in this image, for example, you can see the phase channel in red, and the green is the nuclear dye, which I use for enhancing the segmentation of the cells. Later in the pipeline, with QPI, I can also discern the subcellular structures with high enough magnification. So in this example image, you see fibroblast cells, and the structure is the nucleus with the nucleoli very distinct in the center. And this can be done in live cell setup for up to several days. There are also some specifics for acquisition of this type of microscopy because it's a very unusual way of using light. So there can be no free water surface. And for that reason, I'm using special type of dishes, which are dishes with mid-face plate just above the medium surface, which ensures that the surface is completely flat and the light is passing through perpendicular to the surfaces, and it's not bent or scattered in the sample. The way how one can treat the data from QPI and the way how I analyze the experiments is that I acquire first the time-lapse growth of the cells in different contexts depending on the experiment in the microscope. The data is then exported. If you opt for out of the software analysis, alternatively, you can also do the analysis just in the software. But when I started the project 2 years ago, it appeared to be more convenient to export. So that's the way I do it now. And on high power computing cluster, the images are then merged and cropped together and segmented in cell posts using Python, which is a neural network based segmentation tool. And from those segmented masks, the properties of the individual cells, like their mass or their area or other parameters, can be then extracted, and those can then be plotted and further analyzed. So going back to my research and the research question, what I focus on is cells growing in unlimited fashion, meaning they grow exponentially. So in this example video of a scene, you can see a UTOS human cancer cells growing in the quantitative phase image microscope for around 11 hours, and they are, of course, dividing and moving and growing. And this feature we can also capture by very simple experiments like just counting the cells over time, where we get naturally exponential curve just by doubling the numbers of cells every a day or so. And if we plot the number of doublings over time, we get a straight line, meaning that the cells double by a fixed amount of time. From that, we can calculate the doubling time. In this case, it's about 25 hours. The reason why I'm highlighting this is that there is a very important assumption which is crucial in the approach we are taking, and that is that the average cell composition and overall cellular state is constant. The cells are not changing. They are just growing in number, but their properties are constant, and the same applies to the environment they are in. So we assume that the concentration of nutrients is not changing much throughout the duration of the experiment. And if the cells are present in such a state, we call it non-equilibrium steady state. The steady state part means that the system we are focusing on, the cells here, is not changing anything over time. It's just growing in number, but the composition is the same. But the non-equilibrium part means that there is actually a flux of energy and matter through the system. Even though the cells are on average still the same, they consume something and produce something on the other side. And these fluxes is what I'm interested in, the fluxes of matter and energy through cell metabolism. So to introduce you to this concept a bit, you can imagine exponentially growing cell in a non-equilibrium steady state. And of course, in the metabolism of such a cell, we are talking mammalian cells here, there are hundreds and thousands of reactions going on at the same time. And following these reactions to track all the fluxes of matter and energy would be extremely difficult. But we can focus on the fluxes inside and outside metabolism, which on the inside would be nutrients carrying some chemical energy and having some type of mass. Those could be glucose, oxygen, or any other chemical species. And on the other side, there will be energy leaving the system as waste heat, then a mass as waste molecules like CO2 or lactate or other molecules. And last but not least, the important term of biomass. If we focus on these fluxes in and out, we can basically neglect what is going on in the cell because we capture the overall moves of matter and energy in and out, which cancels out the changes going on inside metabolism. So by focusing on these inputs and outputs, we can easily follow the overall fluxes of matter and energy. To put this into a bit more rigorous terms, we list the chemical species which are entering the metabolism. Those could be the glucose, oxygen, as I mentioned before. And we experimentally measure their physical-chemical parameters. That is the energy they are bearing, denoted as E, and the number of atoms per each chemical species. Atoms of carbon, hydrogen, nitrogen, oxygen, and possibly more. We construct these matrices for all the chemical species and also the species which are leaving the cell, again, with specific set of parameters. And what is neat about such an approach is that then we can basically sum the amount of energy on the left side of this reaction together with the mass on the left side and equal it to the energy on the right side together with the mass. And since, if you remember, there is that law of mass and energy conservation, we should arrive at the identity on the left and right side. Specifically, on the right side, we also have the biomass term, which also comes into play in this reaction. And on this, I'd like to focus now a bit more. But before I do so, let me switch gears a bit and explain why it is important to know the mass of the object, in this case, mass of the cells, and why these specific quantities are important. Let me illustrate that by maybe a bit distant, but very fitting example with a human and the sun. If we take a look on the physical properties of the sun, let's call it metabolism, its power is immensely high, ranging up to 4 times 10 to the power of 26 watts, that is joules per second, and also comparably immense weight of the star. For human, the power would be around 80 watts for the mass of approximately 80 kilograms. So these numbers now might seem incomparable. However, if we normalize the power, that is, if we take the specific power of both objects, we arrive at less than a milliwatt per kilogram for sun and around 1 watt per kilogram for human. So by these terms, one can say that actually human metabolism is way more active than the sun is. And the same applies to the cell. So if we take a look at the specific quantities, that is the fluxes normalized by solar mass, we can talk about specific oxygen consumption in the units of moles of oxygen per second per kilogram, which is the important part. And on the right side, on the west side, there could be specific heat dissipation, that is energy produced by the metabolism in watts per kilogram, or by mass growth, in this case, kilogram per second. And this normalization by mass is very important to understand what is actually going on in a system we study. So in this example, I want to illustrate this case with heat dissipation measured for two different cell lines. Both are human cancer lines, but the details do not matter for now. What you can notice is that the growth curves here in terms of heat are very different. The ACT line grows seemingly exponentially, reaches a peak and then falls down again, whereas the UTOS line kind of reaches just a plateau. So just from this graph, we can't say if this is because the cells grow differently, or if they produce more heat, or if they have more mass, even though their numbers are similar, and then they dissipate differently. But if you take a look at the normalized heat dissipation by the solar mass at the peak value, we can see that actually ACT cells are more metabolically active than the UTOS cells. So in this graph, we see the mass-specific heat dissipation rate in terms of watts per kilogram. So we see that 1 kilogram of ACT cells would dissipate more energy than UTOS cells. Those two systems are growing, and comparing further with non-growing system, we are dissipating even less energy. So just from the specific quantity of heat per kilogram of dry mass, we can really know what is going on in the metabolism of the cell. We can stretch this even further and go from specific metabolic rates in terms of something per kilogram to something per mole, if we know the composition of the biomass, and that is what is the weight of 1 mole of cellular biomass. That we can measure by measuring the elemental composition of a cell, which I have done. And from that, one can then calculate how much, for example, heat is produced by 1 mole of biomass. But for that, the dry mass of the cell is still necessary to be known. So introducing the importance of knowing the mass of the cell, one can also illustrate the utility of QPI by different means, not combining with other measurements, but just looking through the growth curves of different cells in different contexts and seeing what the data have to tell. So, for example, when we explored the role of the seeding density in the experiment, when we started with this methodology, we tried seeding either 50 or 100,000 cells per square centimeter, and we followed their growth over time. So in this graph, you see the average cell mass in picograms over approximately a day. And what you can notice is that initially the two samples are basically identical in terms of average cell mass, which is expected because the cells were taken from the same culture. However, in the more dense sample, the cells reach the plateau much earlier, and they even start to dwindle because they are already tightly packed in the dish, whereas the cells in the less dense sample can grow to higher cell masses in the experiment. The neat thing about QPI is also that we have access to other parameters, not only the cell mass, but also its size and shape. So we can also extract the morphological features of cells in the experiment. So in this graph, from the very same measurement, we can look at the total cell area, and we see that the cells in the more dense sample are covering approximately double the surface and are reaching plateau already sooner than the less dense sample. Another very useful feature, which I heavily rely on, is the capability of QPI to assess the overall physiological state of the cells growing in the cell population. That is important to follow because sometimes the cell might be seemingly normal, but they might have been affected by some unwanted disturbation. For example, they were split too late, or there is contamination in the media. And all these things are reflected in the growth curves, which are measured by a QPI. So here you can compare a growth curve, again, in average cell mass in picograms for either 50,000 cells per square centimeter and two samples with 100,000 cells per square centimeter. If those two experiments or two samples were totally identical, these curves would overlap. But as you can see, there is a shift of the green curve to the left, meaning that these cells were growing more rapidly than the sample here in brown. So probably the sample in the brown was perturbed in some way. Maybe the cells were split in inappropriate time, or were going through some difficulties in the incubator, meaning that one should pay attention to such a sample and investigate further. And before I end the talk, I would like to illustrate another use case we have with QPI, which is being done in collaboration with clinicians, Anna and Angela from the Technische Universität in Dresden. And in this project, we are using QPI to distinguish the differences of cell physiology between cancer-associated and healthy cells. So in this case, we are working with samples from human patients, suffering from prostate cancer. And we have obtained pairs of biopsies from each patient. One sample is healthy fibroblasts, which are not associated with the cancer. And the second biopsy is cancer-associated fibroblasts, which are not themselves directly cancerous, so they are not mutated, and their genome should be identical to the healthy fibroblasts, but their metabolic and physiological state is affected by their proximity to the cancer microenvironment, where physicochemical parameters of the tissue are perturbed. For example, pH is usually lower, and the oxygen pressure is also usually lower or even zero. And by comparing this pair of cells from one patient, we can observe what is the influence of the changed physiological state on metabolism. So in this experiment, we followed the growth of these two cell lines over time. And what is plotted here is the average cell mass in picograms. And you can notice that at the start of the experiment, the cells were basically identical. And only later, there is a difference being pronounced. The healthy cells seem to reach higher mean cell mass values compared to the cancer cells, which is maybe a bit unexpected, and we are still looking into this and trying to figure this out. But our hypothesis, or one of the hypotheses, is that the cancerous cells might rely on the proximity to the microenvironment, and if they are taken out of that context, they are not adapted well, and they don't grow as good as the healthy cells. But this is still open branch in the project. So I hope I could illustrate with my talk that quantitative phase imaging is really a versatile tool, not only in the field of biothermodynamics, but also in clinical applications. And with that, I'd like to thank to all the people involved, starting with my supervisor and the lab, our microscopy facility, which is really great help in setting up all the experiments, the QPI community in the Czech Republic in Brno, and the Telight company, who helped me a lot when I was starting with the microscope, and also our other collaborators from abroad and the funding agencies. And of course, thank you for your attention. And if there are any questions, I will be happy to answer.
Serge,
It looks that you're
Serge,
trying to say
yeah,
something, but we are
you're
muted.
muted. You're muted.
Sorry. Yes, sorry. Yes, I asked if you have questions in the audience. No? So I can begin with one, maybe more for Zuzana, concerning the coherence length of your source and speckle, because I have, on your images, I don't see a lot of speckle. So how do you manage it?
Yeah, this coherence length is ensured by using non-coherent light source. So compared to other QPI systems, which are based on laser, like coherent light illumination, we are working with LED. And this allows us to get rid of all like interfering light coming from the laser interference.
So as a consequence, you have to be very careful between the difference between your sample and your reference sample.
Yes, that's true. Yeah, for the alignment, the reference arm is maybe more complicated compared to the laser-based system, which allows to have much simpler setup of these two optical pathways. But on the other hand, the image quality is much better. And in the microscope, which is like all of the parts of the microscope are motorized, and we have some tools how to keep the microscope tuned. So even if there are some vibration or something in the object arm, we have some like protective mechanism how to keep the coherence still in.
Okay, and because of the interest of holography that you are using, do you use some, maybe more for Vaclav? Do you use the refocus possibility or not? Or it's not necessary.
Could you repeat the question?
Yes, when you use holography, you can also by calculation only, you can change the focus on your image. Do you use it, or is it not useful for you?
Focus during acquisition or
After in post-processing?
That's not a feature I would be using since I'm more interested in like a population or cell population wide characteristics. So I need to actually capture many cells, and in my project I don't care that much about like subcellular features. So maybe this thing would be interesting for people who would be caring about like single cell level, which is not really my case.
Okay.
Yeah.
Do you have questions in the audience?
I just wanted to second what Zuzana said, that even though it might be a bit more difficult to align the microscope and the learning curve is a bit steeper at the start, I myself compared already few modalities of quantitative phase imaging from different vendors as well. And I said that but the Telight microscope one really get unmatched clarity in the image. And once there is a pipeline, it's very straightforward, and the data quality is very high.
In the audience, do you have questions which are more related to biology? Guillaume, yes.
Yeah. Maybe more a question for Zuzana. So when you process the interferogram, I am expecting a reduction in the number of pixels. Is it true?
Yes, it's true.
So what is this reduction? What is this?
Yeah, so currently we have the resolution of 1200, I think 1200, and the original hologram is like not 1200, and the original hologram is I think 2000.
Okay, and maybe another question. So you said you are using a LED, so this is why you don't have any speckle, and you have to ensure the same length, path length in the two arms. But from what I understood, when you do that and you have a tilt, you cannot ensure the same path length from the right to the left side of the camera. Do you see what I mean? And how do you fix this problem?
TIN-L what is tilt?
The reference beam, you have a tilt in front of the camera, right? To generate the fringes. You tilt.
Yeah, you're
right. So how do you ensure the same path length from the right to the left side because of this tilt? This is a common problem in DHM that usually prevents from using a LED. Do you see what I mean?
Yeah, so the tilt is created by the diffraction grating.
Ah,
So
okay, okay. So it fixes the problem. Okay, okay, understand. Thanks.
Is there other questions, maybe relative to biology and dry mass?
Can
Yes.
I ask? Sorry,
Cyril.
should I switch my camera on? No, you don't need it. I have a very naive question about what is the sensitivity towards the refractive index? I mean, when you go to measuring the dry mass, what is the accuracy of the real dry mass you're measuring? Taking into account that you have different thickness, different refractive index in one cell. So how do you manage all this? Because I'm very naive to the technique, so.
Who will respond?
Yeah, maybe you can.
Who can answer that?
I think it's more of a technical question, actually.
It is, but.
That's right.
Yeah, so the phase shift that we are observing depends on the two things, on the refractive
Yes.
index and the
thickness.
thickness of the sample.
Yeah.
Exactly.
That's why I'm asking indeed. So
Cyril, maybe I can give an answer. It has been demonstrated more than 50 years ago. Then for all the components of a cell, you have the same relationship between the optical path, the optical path, and
refractive index.
And the refractive index. So
Okay.
at the end, once you have a chart of your optical thickness, you can convert it to a dry mass.
Okay.
Globally.
Okay.
That's why QPI is so used in this field.
Okay.
It's
got
magic.
it.
It's magic, but
yeah,
but it works.
Okay.
I just found, Cyril, thank you.
Yes.
I already found the slide that I had also like backup. So here are just some equations, and you may see that the dry mass is the equation on the right. The dry mass is calculated from the phase that we did.
Yeah.
And on the bottom, there is something like alpha coefficient.
Yes.
And this alpha is the...
Okay, got it.
It describes the relationship to how the refractive index increases.
Okay.
Alpha
concentration of the things inside.
Alpha is useful for all the cells.
Okay.
All types, except maybe red blood cells, which is a little different.
Because the density is totally different, maybe in a red blood cell. Okay.
Yes.
Okay, got it. So now I promise I will ask a biological question. So Vaclav, in the very last slide you've shown, if I understood correctly what you've shown, you say that the cancer cells are not splitting anymore after a shorter time than the normal cells. Is it what I should understand from your graph, the very last graph? And
I can show it again, actually.
Yeah. Yeah. So the cancer-associated fibroblasts,
the mean cell mass
seems to decrease correctly.
However, there might be more effects going on. It's not that the cell mass is only affected by how well it is coupled between the cell growth and cell dividing. It could be also that the cells are actually getting smaller. We have also seen that in our data in special cases, that the cells are extruding mass. So we are still looking into that and trying to decipher what is actually going on, because this was a very preliminary observation. But it could be one of the effects that the cells don't grow that fast, or meaning that they don't acquire that much of mass over time, or that they change their division rate compared to their growth rate.
So in fact, that was, okay. So maybe it should be interesting to look at how the division rate changes as a function of the mean cell mass. Because as you said, it will give you two types of different information.
Right.
To what I understand, in cancer, you're supposed to have a growing rate which is much faster than normal cells, and when you become malignant, in fact. So I was surprised to see that graph, but as you say, it's not directly correlated to the growth rate. So maybe it's interesting to have a graph regarding the change in the mean cell mass and the growing rate at the same time.
Yeah, right. That's a very good suggestion, and I'm looking into the data, and that would definitely be a thing I should check out.
Okay,
yeah.
Thank you. I have no more questions.
Thanks, Cyril.
Thank you.
Other questions by other people?
Maybe also a naive question, but you apply these techniques only on the 2D cell culture, but can you also apply it in 3D?
You can do that. I think, Zuzka, you had some example videos actually, right, in 3D environment.
Yeah, regarding the 3D samples. So the story is that this technology is a 2D, and what we are recording is like the integration of the signal. So when the light goes through multiple layers of some, for example, organoids or some histological samples, then you would see the phase shift integrated from all these layers. So you couldn't distinguish which layer has what mass. You would just see the black spot. So first condition is this sample still must be transparent, and only it can be like 1 or 2 layers of the cells on the top of each other. Okay. That's the thing. Yeah, we cannot do like these bulky 3D objects or organoids. There you would have to go to, for example, tomography systems.
Okay, or you can also imagine a sample with in 3D, but with a lumen inside the sample, or
yeah, these empty organoids could be something that maybe would work. Yeah, we don't have much experience, as I said. We just started, and we don't have all the applications covered yet. But I had it in the presentation 1 or 2 videos of the cells in 3D metrics, so that might be to getting more to the real conditions in the tissue when you cultivate cells, at least in collagens. It's still single cells or like separated individual cells, but in the 3D scaffold. Mm-hmm. That's something that we can also image with.
Okay, thank you.
Okay, if we don't have any more questions, maybe we can finish now.
Yeah, exactly.
Just before 2 hours, so perfect.
Yes.
So thanks once more. Thanks a lot to Zuzana and Vaclav.
Yeah, thanks a lot. Thanks a lot to also to Serge to have moderate this session. And so, and thanks also for people who are there and regularly present. So the next video conference will be, should be confirmed. It should be the 4th of June. And so all the informations will be sent on the GDR Imabio mailing list. So thanks again, and see you soon. And thanks, Zuzana and Vaclav.
My pleasure.
Thank you for the invitation.